H3H4 these histone molecules, and after the dissociation of these molecules, the finally different RNA transcription or DNA transcription factors, for example RNA polymerase and all those transcription factors, have a clear access to the DNA, and when they have the clear access to the DNA, they can make a they can transcribe the DNA, and they can make proteins out of those mRNAs that they usually produce after making all these things. So DNA histone acetylation is an important step before making any further copies of protein out of this highly stacked interaction. So we have a stacked interaction. When you make a acetylation, we finally end up. So in this case of acetylation, we need different proteins to help this as well, to facilitate this acetylation process, and we have these proteins. One of these proteins is DNA acetyl transferase, or we call it HAT, histone acetyl transferase, we call it HAT. So this HAT proteins is gonna facilitate this reaction process. Okay, and as we can facilitate the production of this nucleosome model from this highly interactor contracted fiber, or the 30 nanometer fiber, we can also make this DNA molecule really tightly packed with this histone protein, so not to opens up, and via the opposite process, and this opposite process is facilitated by another set of proteins. Those proteins are called HDAC or deacetylase. So HDAC, it is called HDAC or deacetylase. So this deacetylase helps them to cleave up those acetyl groups, and to convert them, and to make them again, and to convert them into the extra stacking interaction to make a 30 nanometer loop like that. Okay, and not only the deacetylation, but also methylation can do these things. So in this case, methylation can reduce the transcription process by make those nucleosome model really strongly stacked with each other to make that 30 nanometer fiber. Okay. Okay, now let's move on. Yeah. So we have regulation of transcription inhibitor. Now let's talk about the regulation of transcription initiation, because when we have clear access to the DNA, so it is really highly stacked in this 20 nanometer fiber. From this 20 nanometer fiber, sorry, 30 nanometer fiber, and from this 30 nanometer fiber, when it finally opens up the DNA, what we make a structure like this nucleosome, and finally have access to this free DNA to go on transcription. And to go on this transcription, we need to have different transcription initiation factors, and those factors are needed, and RNA polymers is needed to read that DNA template to finally make new RNA template from it. And for doing this, we have a tight regulation in this case too. So in all the stages we have regulation. Okay, so by making a region of DNA either more or less able to bind with transcription machinery, they can find it. So you can find this organ or the typical structure of eukaryotic gene in this picture. What we have a typical function. So this picture is really important for understanding the eukaryotic genome system. So in this case, what we are having this poly A6. So in this case, what we are having, the most of the time we have exons, which are the functional part, and we have introns, which are the non-functional parts. So we need to cut off all these parts after the mRNA is made, because we cannot cut it from DNA, because sometimes the intron can have those regulatory sequences. The introns can have those heterochromatin regions, which is not made to function. But in some place or in future purposes, that those introns or those regulatory site may be placed on action, and then in those situations we need them to. So in this case, not only they have the structural part of the gene, but we also have the promoter, which is needed for our transcription initiation. And this promoter, we can have those initiation region or Tata box or this concentration sequence, and we also have this Titigaca box where the polymers will bind and then transcribe. And then we also have two different centers for controlling. One is the enhancer, and one is the distal control element, and is the proximal control element. So the proximal control element placed really closer to the promoter, and the distal control elements placed a little bit farther from that promoter and farther upstream for that promoter. And most of the time, those distal control elements are enhancers. and some of those control elements are in the proximal control elements are placed towards the promoter. It is actually called the regulatory regions, because when the regulator proteins can bind, most of the time inducer or inactivator or inhibitor can bind to it and stop the expression of the DNA. So in this case, what we have after all this thing, the final transcript of the mRNA, when it is made, it is except for all these regulatory parts, because this mRNA only is destined for making proteins, and that's why you don't have different control regions like promoter region, or the control regions like enhancer and the inhibition region in this mRNA. So in this mRNA, after the production of this mRNA, we first production of mRNA in the initial stage of transcript of mRNA, which we call the pre-mRNA, is having exons and introns both. But we need to process this mRNA. That's another very important part during the eukaryotic central dogma, that they have to process this mRNA. So they have to process this mRNA with some enzyme they call the spliceosome actually. So they make a complex of spliceosome. So spliceosome is made up with several proteins and several enzymes which work together to finally cut those introns out, and they bring those exons together, they attach those exons together to finally make a coding segment. In this coding segment, what they have a starting point and they have an ending point. So they have the first code which codes for the first amino acid sequence which will start the protein synthesis, and then you'll have the last end of it, we will have a stop codon, and this stop codon makes a protein which will in turn tell the protein synthesis to be stalled. In those cases, the protein synthesis will be stopped. When it starts from this place, it goes through this and it stops here. So it makes the protein. So we finally make a region of compact protein structure after deletion of this introns from the exon region by doing this. And not only by the modification of this structural region or the protein coding region, it also have a simple modification in the 5'. It actually add guanosine triphosphate, and this is slightly modified guanosine triphosphate in those side in the 5' region, and in the 3' region it attaches many adenosine triphosphates, many adenosines to attach each other to make a polyadenosine tail. And this modification helps this mRNA to derive or to make a journey from the nucleus into the cytoplasm via nuclear pore of the nuclear envelope. Okay, so that's how mRNA is processed, and after this mRNA processing, it will be translated to make proteins. So in normal these cases of an activator, in case of this production of mRNA from a DNA, what is the function of the enhancers? As we have as I have discussed, the enhancer can be found in distal control elements. So these enhancers will be simple protein molecules. When they bind to the enhancer region, they are placed in a really upstream position with from this starting point or the promoter site of the DNA transcription. So they need to make themselves really available from the promoter or the DNA or RNA transcription factors to take on. So that's why they bend themselves together like this. So they have a bending DNA protein act on these places. So the DNA bending protein, and when the DNA bending protein comes in, it helps the DNA to bend. So when the DNA bends from a straight line to the curved structure like that, it will actually place this activator site with this group of mediator proteins, and when these proteins are together with a complex with promoter, protein complex of transcription initiation and the enhancer, it gives a signal to the cell to go on. So RNA polymerase comes in and it starts the RNA synthesis. That's how the RNA synthesis can begin. So what we are looking at in these cases is coordinately controlled genes. So we are not talking about simple operon things. When one gene, when several genes are situated one by another, and one regulatory site is there to control the whole expression of those different genes. So it's a regulatory site. But in this case, eukaryotic genes, what we are looking at this gene, and this gene will be controlled by only one regulator. So one gene controls one regulator in this case. So one regulator for that, and one enhancer in this case. So what is gonna do? It is gonna actually bend like this to make this function. So we have one enhancer, we have inhibitor. So when the enhancer is signaling, the transcription will on, and when the repressor is signaling, the transcription will be halted, or transcription signal will get off or cut off. So this is the system. And let's talk about the post-transcriptional regulation. That's another important thing about the eukaryotic genome that cannot be found in case of prokaryotic genome, that after the transcription of this gene, after the transcription of DNA, when we make RNA, those RNA can be, or those transcript of RNA can be controlled. So during this alternative RNA splicing, what we can do, we can do different alternation in this splicing. So we have talked about the RNA splicing, but in this RNA splicing event, that event cannot be occurred as simply as I have discussed. But in this case, that is doing some different job. So what is the job? That the whole part of the gene is not expressed, it is not making one mRNA. Instead, it can go through two different ways to make two different kinds of RNAs. So they have several exons and those introns, so they need to leave those introns. So finally what they can make, they can add those exons together. So they add those exons to make a protein like this. So, and also they can arrange those exons to make a protein, like this. So how? And so if they make this mRNA, that will make protein 1. So, and suppose this will make another mRNA that will make protein 2. For example, when they make this protein 1, this protein 1 is a kinase, for example. What kinase do? Kinase is actually phosphorylating different things. And when it makes protein 2, it makes phosphoprotein phosphatase. So what phosphatase is going to be going to do? The phosphatase is going to cleave off phosphate group from different proteins molecules. So they are acting differently when they are expressed. So that's why they are coming from a same gene. So why? So that's why we have to have a really tight regulation or control to make those genes. If we have to access or to make a signal to finally make this protein or protein 1, they'll make a kinase. So when they needs to break down, when they needs to have a signal system into the cell to phosphorylate different protein molecules for different further purposes, they will turn on to production of this mRNA, and that's why the kinase will be produced. And when they have to break down some things, for example, in the case of from making glucose from glycogen, in this case they need phosphatase. So this phosphoprotein phosphatase will be needed. So in those cases, they triggers the synthesis of this kind of mRNA, production of this kind of mRNA. So in those cases, the synthesis of mRNA have no difference. So there is the normal transcription going on, that synthesis of mRNA is done, but the difference is during the splicing. So the RNA splicing is altering in all these cases. So not only the regulation can be placed during this transcription, but also it can be placed during the mRNA splicing. Okay, and now let's talk about the mRNA degradation. So when the mRNA function is over, so the concentration of mRNA is getting higher in the cell cytoplasm, and cell do not want different RNA genetic materials, for example different nucleic acid materials in its cytoplasm. That's why they need to degrade all these parts. So when the work is done, they need to degrade this RNA. So we have these hydrogens which is normally so we have mRNA which have these bases, and those bases can have different complementarity of each other. So those bases are normally, when put together, they can arrange themselves like this. They can arrange themselves to make a structure like this. So for example, they finally can make different structure like this loop-like structure, because they cannot stay on its own like this previously. So they'll make this loop-like structure, and when they make this loop-like structure, another set of enzyme which is called dicer enzyme. So dicer enzyme comes in and they clips off the header portion of that loop-like structure. So they'll break this header portion and take this header portion away, and not only that makes and cleave this header portion, but they can also cleaves out simple regions among those DNA hybrid. In those DNA hybrid, they'll cleave this simple portion and finally makes different part of the DNA. We call it the micro DNA, because these DNA samples are really smaller. So those are not, sorry for that. So it's not DNA. It's a pretty mistake. So whenever talking about two strands, we're looking at DNAs, but it's not DNA, it's mRNA, because mRNA is has made like such a way to make a structure like that. So these are called the micro mRNAs or miRNAs. So these micro RNAs are really plays a key role for this transcriptional purposes. So some of those micro RNAs can have a protein complex attached to them, and then those micro RNAs can be targeted into some mRNA. So when the protein complex bind to that micro RNA, it will be targeted to to different mRNAs. So now when they are targeted to attach with mRNA, they will bind with mRNAs. As they are very small, or there is the 4, 5, 6 nucleotide long, so there is a higher probability of binding with other mRNAs, mRNA bases. So they bind with different mRNAs here, and they stall the synthesis of protein via the different ribosomes to make proteins in those. So in this case, what they are doing, they are actually preventing those mRNA to be translated by doing this. So these mRNAs, these miRNAs, or the microRNAs, are actually interfering with the protein synthesis. So that can be done via this mRNA degradation step that can block this translation reaction. Okay, but if we're talking about normal mRNA scheme and normal mRNA translation scheme, then what we can find a normal process of going to translation. To go through this translation, we have to release those blocked site of the regulatory proteins. So in this case, there is a regulation by doing this protein complexes. So when we have to synthesize some proteins, so then we have to stop the production of this complex protein that will gonna make this microRNAs to bind with our mRNAs. So in those cases, the production will be halted of this protein. That's why those mRNAs cannot be bound with this with the other mRNAs, and that's why the synthesis by coding this protein, coding those protein can be easily derived through translation procedure, and that's how the translation can be go on. Okay. So that's why the concentration of mRNA in the cellular cytoplasm is really important. So the cytoplasm is really important in those cases, because the mRNA concentration in the cytoplasm, if it is higher, in all those cases, what can happen, then those mRNAs can make devastating effects, because those mRNA can bind with other mRNAs to make duplexes of that. Those mRNAs can be chewed off into small parts, as we have seen, that can interact with the production of the protein and all these things. So that's why it's important to have that, and we can have the protein processing and degradation. So what is that? Now let's talk about the after translation or post-translational regulation scheme. So we are not only having a transcriptional regulation, we are also having the post-transcriptional regulation or RNA splicing regulation, and then we will have a translational regulation. Finally, what we are having is the post-translational regulation, and in this regulation, what we have after the making of a protein. So when the protein is made, so suppose this is an endoplasmic reticulum, we have this ribosomes doing on here, and we have made some proteins in this case. So for example, it makes some proteins. So after making some protein, we have end up with this like this. So protein is inside the endoplasmic reticulum. Let's draw the endoplasmic reticulum here. This is the endoplasmic reticulum with this protein, and we finally have the Golgi bodies. So in this Golgi body state, we can have this protein, and finally it can pinch those things off like this in the vesicles, and finally what we have, cell membrane, and this vesicle is going to fuse with that membrane, and finally have the release of that protein. So suppose we need to discuss about the release of a protein from after the synthesis. So when the protein is made, it is in the endoplasmic reticulum. So this is ER, and from this ER the protein makes a journey as a packet of bulge towards the Golgi bodies. It makes a journey towards the Golgi bodies. It makes cis Golgi, then goes to intermembrane Golgi, and then finally trans Golgi, and it cleaves off via like the vesicles in the Golgi bodies. In this big G, on the Golgi bodies here, so in this Golgi complex, it actually modifies. So we have the protein, and the protein is further modified. So the protein is here, and the protein is modified, and after the modification of the protein, suppose this is the modification, and most of the time the protein is attached to two different mannose phosphate subunit like that and all these things to modify the protein, and actually it makes a tag, or it attach tags to deliver these proteins into different regions of the cells. For example, in this case the protein has to be delivered in the outside the cell. That's why you need to fuse with the cell membrane. In some time when it needs to deliver that protein into an organelle like mitochondria, then it tags with another set of proteins, and that helps them to go towards the mitochondria and different things like that. And so after doing all these things, what they can do, they can actually sort these proteins to go in different directions. So it will make to go in different directions. And after this result, so there is a controlling system going on in these Golgi bodies. They're signaling of where the protein is going to be, and after the signal is complete, and the protein is destined to deliver in that particular region. For example, now in this case it destined to deliver here. So we have different modification, different proteins, and this protein is finally going to release outside the cell. So that's how a protein is made and going outside. So this is post-translational modification of cell or the protein sorting, which is going on inside the Golgi bodies after the production of the protein in endoplasmic reticulum. And when the protein production is needed and when the protein is not needed, and most of the time when we encounter some of the junk proteins which is under the cell or inside the cell cytoplasm, we need to